Thank you for that kind introduction. My name is Edward Alec Pajarillo. I'm a PhD candidate at Dankook University. Today, a pleasant afternoon to everyone. I'm going to talk about the effects of weaning, host breed, and cohabitation in the swine distal gut microbiota. So my presentation this afternoon will be subdivided into the background, the study and the methods, and my results. Pigs are important in agriculture, industry, and environmental sustainability. They are one of the most important food commodity for us humans. Do you recognize this picture? This is a picture of the most famous Korean dish known as samgyeopsal. And not only that, pigs are now considered as a replacement for mouse and rodents in human-related studies. So it's really important to know the factors that affects the physiology and gastrointestinal tract of these animals. The gastrointestinal tract of pigs is home to trillions of microorganisms and over 1,000 bacterial species. So outnumbering the cells in our body and the genes, it only implies that these microorganisms are not only important in metabolism, but also in production of nutrients, recycling of minerals, and the development of the immune system. So identifying and characterizing the microbiota will help us understand the dynamics of these microbial populations in the gut. Now to the design and method. DNAs are everywhere, and this machine helps us detect and quantify into a huge library of sequences so that we can analyze the microbiome of the animals. So we can use the three major components, the adapter, barcodes, and the primer sequence. The primer sequence is used as a specific to our target DNA, which I focus on the 16S rRNA gene. 16S rRNA gene are ubiquitous or present in all bacteria. So to be particular, I focus on the V1 to V3 hypervariable regions of that 16S gene. So this is widely used in many studies. My first study focused on the effects of weaning. So weaning is a normal event in animals and humans. So it involves the separation of the piglet from the mother, as well as the shift of diet from liquid milk to solid feeds. So I want to know and characterize the microbiome differences in piglets before and after weaning. So as you can see in this slide, that there are really differences in the microbial. Those changes are detected at the phylum level. Here you can see that Firmicutes is highly abundant in pre-weaning or before weaning, and Bacteroidetes is highly abundant after weaning. So we can observe this in detail further down the taxonomic levels. So in this figure, you can see the differences between the microbiome of pre-weaning pigs and post-weaning pigs. So I want to focus on two major bacterial genera, which is the Bacteroides in pre-weaning or before weaning and Prevotella after weaning. So these two bacteria, actually they are from the same phylum, the Bacteroidetes, but they have differences in functionality. Bacteroidetes, for that matter, is highly associated with monosaccharides or oligosaccharides in milk, while Prevotella is highly associated with the polysaccharides or hemicellulose in a plant-based feed diet. So definitely, those differences in a microbiota would mean that they have differences in gut functionality. The next study is determining if there are differences in the microbiome of three purebred lines of pigs. Host genetics, as we know, are controlling gut-related functions and diseases. This is widely known in humans. And in my study, I used Duroc, Yorkshire, and Landrace, which they have different performance. For example, Duroc is known for their high growth capacity. Yorkshire is known for their maternal maternal abilities, and Landrace is known for their reproductive capacity. So host genetic controls phenotypic characteristics, and also those characteristics can be manifested in the breeds. So in a previous study, using genetic markers, these three breeds are actually different. And in our study, the microbiota of these three purebred pigs experience or showed the same trend. So we plotted in a principal component analysis individual microbial communities of these pigs to verify if they are really distinct from one another. So as you can see, Duroc, Yorkshire, and Landrace are separated from one another, particularly the distinction of Duroc in among those two breeds, the Yorkshire and the Landrace. So these differences can be found or can be detected in this heat map that I'm showing right now of the bacterial genera. So here I'm showing the unique bacterial genera in each breed. So for Duroc, Landrace, and Yorkshire, these are the bacteria that are unique for them. So they may have functional or disease-related implications for these three breeds that needs to be studied. So in my last study, I determined the effects of cohabitation. Cohabitation is a normal practice in animal husbandry. It involves the mixing of litters or individuals in the same pen. So it increases interaction, it increases aggression, and sometimes the risks for exchanging pathogenic bacteria can also happen in this situation. So the three breeds, the Duroc, Landrace, and the Yorkshire, were cohabitated for 0, 6, and 12-week period. And we determined those differences in the microbiota. So in alpha diversity, we found that at week 0, the alpha diversity values of these pigs are less uniform compared to the alpha diversity after cohabitation at week 12. So longer cohabitation means that pigs exchange bacteria through time. No new bacteria are being added, but the ones that are existing in the community are being exchanged with one another. So in this PCA plot, we can see that at week 0, the enclosure is larger than the enclosure at week 12. So it only shows us that the longer the cohabitation, the similarity increases in the microbial composition. But that's not the end of the story. We use 3 purebred pig lines, right? So we want to know if there are still host specificity or breed specificity in their microbiome. And true enough, we found that even we cohabitated them, at any time point, the microbiome or the microbiota of pigs are still distinct with between Duroc, Yorkshire, and Landrace. So we believe that still host genetics has some influence in the microbiome, in shaping the microbiome of pigs. And there are bacterial genera that are actually distinguishing among these three breeds even after cohabitation, particularly Dialister and Bacteria. So we are wondering what are the possible implications in health or in disease for these three breeds of pigs. So stress and diet in weaning is shaping the microbial composition of pigs at weaning. So this is very important because now we can know how to control the negative effects of weaning. As we saw in previous slides, that host genetics can shape the microbiota of pigs, and cohabitation increases the similarity of their composition. And there are specific bacterial genera that we can study for their possible functional or disease-related implications in pigs. So with that, I'd like to thank my professor, Dae-Kyung Kang, of the Laboratory of Industrial Microbiology, and the members of our lab. So for future references, please see these publications. Thank you very much for your attention.
시간 굉장히 잘 지켜주시는데 발표 부분에 대한 질문 있으면 질문해 주시기 바랍니다.
한 실험마다 토탈 리드 수가 어떻게 되나요? What is the total read in each experiment?
In each experiment, I can only give you an estimate, not the exact value, but it's more than 100,000. More than 100,000.
제가 질문할게요. 질문을 하자면 what do you expect the reason the cohabitation experiment? What do you expect the reason for if the pigs are co-sharing cohabitat, and what do you expect the reason they share this microbiome after like 10, 12 weeks?
The increasing interaction of these pigs inside the pen, that increased interaction from mouth or from fecal
Are they
matter.
sharing like their food or like water?
There are many factors actually, but yes, food is there, the water, but we cannot neglect the fact that these pigs are in contact physically with one another, so they can, the mouth of the pigs can go in front of another pig and or in the fecal excrement, like that. So there are many possible factors, but cohabitation really increases similarity of the microbiota. There are just many factors to consider.
Thank you for your answer. 그러면은 첫 번째